I'm Hermann Zellner, software engineer in the UniProt team at the European Bioinformatics Institute. And in this webinar, I would like to introduce you to the software package UniFire that allows the users to automatically generate functional predictions for their own proteins. So why do we need automatic annotation, automatic function prediction for proteins? Due to the advances in sequencing technologies, an ever-growing amount of protein sequences is available. However, we can only annotate a small number of these sequences manually. And therefore, in UniProt KB database, we are using methods that infer the function of unknown protein sequences from proteins with known functions. The methods that we use are based on criteria like sequence similarity, protein families and domains, taxonomy, genomic context, and so on. And by combining these criteria, we have created annotation rules for protein sequences. We have realized that not only the protein annotations, but also the annotation rules themselves are a valuable knowledge, and this knowledge should be shared with the public community. And therefore, we are making our annotation rules available to the public. This will open up UniProt automatic annotation systems to the community and facilitate the exchange of annotation rules between different research groups. In order to do this, we are providing a way to apply on your own data, to apply the rules on your own data as a standalone tool, or maybe in future also through a web service. The source code of the project is available in EBI GitLab repository to the public. So the project is also open for contribution, and we would also like to ask for your feedback about it. So we have generated also a mailing list where you can register and ask questions about the software, get in contact with us. We are going to announce on the webinar new features, and if you would like to bring the project forward, then please get in contact with us. So I would like to give an overview over the functional annotation systems that we use in UniProt to annotate UniProt KB. So first, UniProt KB is composed of two different sections. The first section is the reviewed section, also known as UniProt KB SwissProt, which is annotated by manual curation from the literature. Its function for the rule generation is a reference for quality control of the rules. The second section is the unreviewed section, also known as UniProt KB Tremble. And this is the section that is automatically annotated by the rule systems. So we have two different rule systems in place to annotate UniProt KB Tremble. The first system is called UniRule. It is a semi-automated system, which means that human curators are responsible for creating and maintaining the rules. You can see the UniRule workflow in the diagram on the top left of the slide. Curators are continuously creating with every release new annotation rules, which are compared and verified against manually curated annotations in the reviewed part of UniProt KB. There, a rule must show high accuracy when applied to SwissProt before it is released to annotate the unreviewed part of UniProt KB Tremble. Currently, it's about 30% of all unreviewed proteins from UniProt KB Tremble that are annotated by UniRule rules. These rules provide various kinds of annotations like protein names, gene names, function comments, comments about catalytic activity, or a role in a pathway. Also, position-specific sequence features like active sites or binding sites are annotated by UniRule. You can find all UniRule rules on UniProt website. At the bottom of the slide, you can see the representation of an example rule on UniProt website. You can see that a rule consists of two parts, conditions on the left and annotations on the right. So if the conditions of a rule are satisfied by a certain protein, then the annotations in the rule are predicted for that protein. In the case of this example rule, if a protein is matched by these two signatures, then those annotations on the right are predicted for that protein. A more complex UniRule is shown in the next slide. So the logic here is a bit more complex in order to be able to predict more specific annotations. For this example, we have, excuse me, for this example, we have a main part with a signature condition and a generic taxonomy condition for bacteria. If the generic condition is satisfied by the protein, then some generic annotations are predicted for the protein. Additionally, we have a more specific taxonomic condition, Enterobacterialis. If this condition is satisfied by a protein, then some more specific annotations will be predicted for the protein as well. And on top of that, we have in this rule some pattern conditions. In this case, the positions of certain features are mapped from a template protein to the target protein through a sequence alignment. And afterwards, the pattern is checked against the target sequence interval. And if the pattern is found on the target protein, then the additional annotations will be propagated. So far about UniRule, the second system that we are using to annotate unreviewed proteins in UniProt KB is called SAS, which means Statistical Automatic Annotation System. It is a fully automated system using the C4.2 decision tree algorithm. Based on the condition types, interpro signature match, sequence length, and taxonomy, it learns automatically high confidence rules from the reviewed proteins in UniProt KB SwissProt. These rules are then applied to the unreviewed proteins in UniProt KB Tremble, which gives automatically annotated protein records in the end. As you can see in this example, the structure of a SAS rule is simpler than the structure of a UniRule. A SAS rule can only have one set of conditions and one annotation, other than for UniRule rules. If all the conditions in the set of this SAS rule are met by a protein, then the one annotation is predicted for that protein. So to summarize, the rules from both systems, UniRule and SAS, are composed of two parts, conditions and annotation. If the conditions are met, then the annotations will be predicted for a protein. In UniRule, we use the following conditions: matches to interpro signatures, taxonomic conditions, or scientific organism name. We can also filter by a gene location like organelle, or by sequence criteria like sequence length, or if the sequence is a fragment, or if the sequence follows a certain pattern. The predicted annotations can be of type protein name, protein function, gene ontology term, or catalytic activity that a certain enzyme has, the pathway membership, or subcellular location. In case of UniRule, that can even be a specific positional annotation, like where exactly an active site or a binding site of the protein is located. So now that I've introduced our rule system, we would like to make these rule systems open to the public. So in order to do this, we require two components. The first component that we need is a file format in which we can represent our rules and exchange them with the public. This exchange format should use well-known standards and be well supported and understood. It should also expose explicitly all the logic that is necessary to interpret the rules. And the second component that we need is a software engine which allows to apply the rules on a set of protein data and retrieve the inferred annotations. For the first component, we have designed a new format to represent protein annotation rules. We've called the format the UniProt rule markup language, or short URML, and it is based on XML, which is a well understood standard. The logical unit in this XML format is a rule which is again composed of conditions and annotations. It is also linked to a fact model which can hold protein facts like protein sequence, taxonomy, or matches to interpro signatures. At the bottom of the slide, you can see the general structure of the format. You can see that the rule schema is linked to a fact schema, which I'm going to describe later on. In UniRule, one UniRule is represented by more than one URML rules, because a URML rule is a simpler structure than a UniRule. And therefore, each URML rule contains an element group, which allows to group together different URML rules that belong to one UniRule. So in the next slide, you see the fact model, which I have mentioned before. It can represent protein data and protein-related data, like, for example, matches to interpro signatures or the taxonomy of a protein. And it can also hold data like a protein name or a gene name. As we have seen before, the fact model is used inside the URML format to represent the rules. In the fact model, you can recognize objects that are used in UniRule and SAS rules. There are objects like the protein name or gene name, which can be predicted by the rules. And you can also find the taxonomic lineage or protein signatures, which are used as conditions in the rules. As you can see in the slide, these objects are linked together. Overall, they are sufficient to represent all input and output data of UniRule and SAS rules. So they are not only used inside the rules, but also they can represent the output data, so the protein annotations that result from the rule and the protein data. So let's have a closer look at how we represent the rules in the URML format. In the next slide, you can see an example of the URML format that shows the condition part of one rule. In this slide, you see two different blocks, in this case, two AND blocks, where each block contains several conditions and their logic is combined by an AND logic. The two blocks are again separated with an OR logic. So let's have a closer look at the second block in order to explain the expressiveness of this format. So if you look at the second block here, if we read through that, it means if there is a fact that we have called protein, so the protein is a field, and that fact is having an attribute sequence with an attribute is fragment that is false. And then it's combined with AND logic to the next condition. That condition says the organism of the protein has an attribute lineage, and that contains the taxonomy ID 2157, which stands for archaea. And then it is combined with analogic with the third condition, which means there is a protein signature with our protein, so linked to our protein. And this protein signature has an attribute signature of the type HAMAP. And the name of this signature is MF0124A, which is the identifier of the HAMAP signature. So you can see this URML format exposes explicitly the logic of a rule. So far to the condition space, let's have a look at the action space, at the annotations that this rule is giving. On the next slide, you see the action space of the same rule, which specifies the annotations that are triggered if the conditions are met. Here we create as a result protein annotation objects with a type and a value. For example, we create here an annotation of type gene name primary and of the value MOAC. So this is basically the gene name that is predicted by this rule. You can see that here in this annotation, for example. The created protein annotations are linked to the specific protein and to the evidence of the rule in the action field. This allows us to keep the link between the predicted annotation, the protein, and the rule which has triggered the annotation. So I have explained about the format in which we can represent protein annotation rules. We have also created a software engine that is called UniFire, which stands for UniProt Functional Annotation Inference Rule Engine. The purpose of this engine is to create predictions for new protein sequences from the provided rules. On this slide, you can see a typical workflow for UniFire software. This workflow fits the generic situation that you have some unknown raw protein sequences and you would like to generate functional predictions for these protein sequences. The first step is to prepare the protein sequences in multi-FASTA format, which is well-known standard. The headers in the multi-FASTA file need to contain the full taxonomic lineage of the organism, but you don't need to collect this full taxonomic lineage yourself. We are providing a script which helps to fetch the full lineage for a certain tax ID. The next step is then to run InterProScan on the multi-FASTA file. You can do this either through the InterProScan web interface that I will show later on, or locally, depending on the number of protein sequences that you would like to annotate. Afterwards, the output of InterProScan is used together with a rule file as an input to UniFire. If you are using UniRule rules, you have the option to provide template alignments as an additional input. The template alignments are provided each release by us together with UniRule and SAS rules. These template alignments will allow the prediction of position-specific annotations like active sites or binding sites. As an output of UniFire, you will get predictions of functional annotations for your proteins in a tab-separated file format, or you can even choose to get it in a FACT XML format. The source code of UniFire is available in our public GitLab repository. I will demonstrate in the last part of the webinar how you can download it from there, how you can build it, and how you can use the code in the repository in order to annotate your own proteins. The repository is also open for contribution, not only for to provide you the software. If you would like to participate in the project, then please send me an email. My email is shown on the first title slide of this presentation and will be available online after the webinar. The GitLab repository also contains a wiki in which you will find news about the project, like new features that we have implemented. And you can also download the latest SAS and UniRule rules together with the template alignments from our FTP site that is shown on the next slide. So now we are coming to the last part of the webinar, where I will demonstrate how you can use Unifier on your local computer. First, I will show how you can clone the Unifier repository to your own machine, and then I will demonstrate how to build it and how to prepare your input protein sequence data, and finally, how to run Unifier software and generate predictions for your own protein. So I'm switching to the console now. So the first step I'm going to show is how to clone the repository. So I've prepared the command on the console. So you need to have Git installed on your machine, and you can simply clone the repository with git clone. So it will generate then. It's not too big, so it doesn't take too long. So it has generated now the repository folder Unifier here, and we cd into it. And let's see what is inside. So these are the submodules of Unifier, and there is a script called build.sh, which is, as the name says, used to build the repository and afterwards also to download the latest SAS and UniRule files in URML format from our FTP. So I'm just executing the build script. On this computer, it will be quite fast because I've done it before, and there it has downloaded all the Java dependencies that are necessary to build the source code. So it doesn't need to do that again. If you are doing it the first time on your machine, it may take, depending on your internet connection, a few minutes, but it's faster here. So the build is already, it's almost done now. It is downloading the SAS and UniRule files from the FTP. And that is it. So now the Unifier software has been built and is ready to go. So the next step is that we need to prepare our input faster data. So I have prepared here in another folder a FASTA file, which is basically one protein sequence. Just a bit about the background of this sequence. I wanted to show Unifier on a new protein sequence that is not available in any protein database, and therefore I have chosen an artificially created protein sequence that is created by ancestral sequence reconstruction for a certain enzyme, his F in his F synthesis pathway. So this is basically an artificial his F protein that is not available in any database. What I have added into the FASTA header in this format is the tax ID 2, a very generic tax ID for bacteria, and additionally some organism name. So as I've explained before, the first step is to fetch the whole lineage, and for this, inside the Unifier repository, there is a script. So I'm going to the Unifier repository that I have cloned before, and there is a script fetch lineage. I'm using the remote one because this is using a remote service, and we only have one protein for which we need to fetch the lineage, so it will be fast enough. So, and I want to fetch it for my input file mystery fastA. And the second argument is the name of the output file. So if I execute this, there should now be a second output file mystery lineage fastA. And it contains now in the header, additionally to the tax ID 2, also the root tax ID 1 and the tax ID for all cellular organisms. So bacteria is already quite generic, therefore the lineage is quite short in this case. So the next step is to use this fastA data to do interpro scan. Because we have only one protein where we want to do interpro scan on, we can use the web interface of interpro scan that you find under this URL here. So I copy and paste this fastA into the browser and submit the job. So this will now take a few minutes to run, maybe 2-3 minutes. In the meantime, so we don't have to wait, maybe you've seen I have already another file here, which is called mysterylineage.xml. It is, I've done basically interpro scan before, and it is the output of interpro scan that I can show you now. So the output of interpro scan contains the protein sequence. It also contains in this element here the previous fastA header from the fastA file. So we are keeping our lineage that we have downloaded before. And the most important part from interpro scan, we have the matches to all the interpro signatures in this file. So this output of interpro scan is now used as an input to Unifier. So to run now, maybe we can have in the meantime a look at our interpro scan is still ongoing. I can go back to this and show you where you can download the interpro scan result. So to run Unifier, I'm going back now to the Unifier repository, and we are providing the unifier.sh script in order to run the software. So if I just run this command without any arguments, it will print me the usage. So we can put together now the command that we want to run. The first command, the first option is I, where I specify now which input file, so which interpro scan input file I would like to put there. So this is my mystery lineage.xml. The second input that we need is the rules file. And as I said, when I've executed the build.sh script, it has also downloaded the latest unirule and SAS rules. So these unirule and SAS rules are found now under the repository in the samples directory, and they are there as unirule URML latest XML. As I'm using now the unirule set of rules, I'm also adding the optional argument -t to add the template alignments for unirule template proteins. This will then allow us to predict position-specific annotations. We need the template alignments for mapping certain positions from a template protein sequence to the target protein sequence in order to predict position-specific annotations. So these alignments have also been downloaded into the samples folder, where you can find them as unirule templates latest. And now I'm checking. The last argument that you need, of course, is -o to specify your output file. So let's call the output file mystery annotations. So by default, the output format will be tab separated file. If you prefer to get as output format fact model XML, then you can specify that with the -f option. If you specify -f XML, it will print it in XML format. Also, the default input format is InterProScan XML. If you have prepared your data in fact model XML format, then you have also a switch, which is -s option. So then you can add -x XML if you want to input your data in fact model XML, or you can even input your data in UniProt XML format. But the default is InterProScan XML. And just as a last bit, you don't need to do that if you're running it yourself, but I would like to start this job with low priority in order to not disturb the webinar. So the main CPU priority on the CPU is given to the webinar, not to this job. And then we are ready to go. So this will now run for maybe 10 minutes. It's printing at the beginning the inputs that I've given and the different options. Maybe in the meantime, while it is running, we can look back at the InterProScan. So InterProScan has finished in the meantime, and this here, so you can see displayed on Interpro website different matches to Interpro signatures that are given. But the important part for Unifier is that you can download on the export button the result of input of InterProScan, and you can download it as an XML here. And this will then be the InterProScan XML that you use as an input to Unifier. So while Unifier is executing all the uni rules, maybe we can go back to the slides, because just in case this demonstration should fail for any reason, I have prepared some backup slides. So we have the first slide is showing the input sequence that we are using. I've added the reference to the publication where I've taken it from. And what you can do also is a blast search to this input sequence, and you will find for the best hits a sequence identity of 76%, which is not enough to infer exact annotations by similarity from this. So basically what I've done is run InterProScan on this FastA file, which will give me the matches in InterProScan XML format. Here we also had a look at the Interpro XML output. I've given you in this slide the URL where you can run InterProScan on the web service if you don't want to install it locally. And then I'm giving the commands how you can clone the Unifier repository and how you can build it. And in the end, I'm giving the command here how to run Unifier on all the input data. It shows the log output and it shows the result that you can retrieve in two different formats. First format that we have done is tab separated. I've opened it up here in Excel. And the second format is the fact model. So let's go back to our console. So you see that Unifier has finished and it has given us this output file, which basically contains the same annotations that you have seen before on the slide in tab separated format. So to have a closer look at these annotations, let's go back to the slide because they are nicely colored in there. So for this artificial protein, we got on the top, it shows maybe the most important annotation, the catalytic activity reaction for this protein. It is now in the new format that was introduced about a year ago into UniProt. It contains, apart from text that describes the complete chemical reaction that the enzyme is catalyzing, it contains also references to RIA database, where you can find under this identifier the reaction, more information about the reaction that this protein describes. It also contains it contains references to identifiers of the reactant in KEVI database or in RIA database. And on top of that, it also contains the EC number for this enzyme activity. Furthermore, it has predicted a text description of the function of this protein. So it catalyzes IGPS conversion of PRFA, which basically describes the reaction in nice words. It annotates the membership to amino acid biosynthesis pathway, and it annotates the recommended full protein name, which is imidazole glycerol phosphate synthase subunit is F, because this enzyme is basically a homodimeric enzyme, and we have and this protein sequences belongs to the his F subunit of this homodimer. Also, it predicts 3 Go terms for this protein, and because we have given the template alignments as input to Unifier, it also gives 2 position-specific annotations for this protein, which are the 2 active site residues on position 11 and 130. The same annotation, the same predictions, you can also retrieve in fact XML format as it is shown on the left, if you prefer that format. So to summarize, or let's have another look at the exact rule, how you can find the rule that has triggered most of these annotations. So when we go back to the previous slide, most of these annotations were predicted by a unirule UR 301401. And with this identifier, you can find the triggering rule on unirule website, which where you can get through uniprot.org on that unirule website. And there, if you search for that rule, you will find that it's a rule for bacteria and archaea. We have given the taxonomy ID for bacteria, so that is why the rule has been triggered. And on the right side, you will find all the annotations that have been predicted for this protein. Also, you find some special conditions that were triggering the position-specific annotations for the protein. To summarize, so we would like to exchange our rules for protein functional annotation with the community. And for this purpose, we have created a file format to represent our rules. And we have also created a software engine that uses those rules and creates functional predictions for new proteins. So what we would like to get now is your feedback and your ideas about the project. So we'd like feedback on the rule format, on the input or output data format, but also on the engine itself that runs the rules. So we would also welcome feedback how you are using the tool for which projects you are using it. So, and if you want to contribute to the project, then please send me an email and we can generate an account on the GitLab repository. So you can even push some feature code that you would like to be added to the code base, and you can generate some pull requests so we can merge your code into the master branch of this project. The question is, what are the rules used to select 3D protein structure associated to any particular gene name? So the rules in UniRule and SAS are basically the rules that annotate the UniProt KB database, which is a protein sequence database. So this database, UniProt KB, links via cross-references against PDB as a three-dimensional protein structure database, but the rules themselves use sequence features only. So the rules themselves use interpro matches in order to identify certain patterns in the sequence. My protein is not well annotated on UniProt, so not getting detailed annotation from UniProt reviewed database. So if so that depends on the fact if the protein is part of the reviewed set of UniProt KB, then it should have all the information that is available in literature because it has been curated by the experts. If the protein is available in the unreviewed section of the database, then our rule systems have the job to annotate it, and the coverage is of the unreviewed section is good, but we are continuously working on improving this. So it's if in the case the protein is in the unreviewed section and it has so far not been annotated yet by our rule systems, then maybe in future it will be annotated. Then there is a question, what about polymorphic variations if it is associated with the available entry? How does it get incorporated? So polymorphic variations are only in the reviewed set of UniProt KB incorporated into an entry. So so let's say in SwissProt you have one in the reviewed set in SwissProt you have one protein record for one gene, which means this protein record will contain all variation data. In Tremble, if you have several genomic sequences and several protein sequences for the same protein with a slight variation, then in Tremble, in the unreviewed set, you will find different proteins. And both different protein records will then be targeted by our automatic annotation systems. So if the variation is not too strongly affecting our automatic annotation system, so if the variation is not at, for example, an active site residue, then the two proteins should get the same annotations.
Ah, sorry, it's Alistair here. We got unmuted.
Yes, you should.
I'm off the chat box. I was answering a question about what to do about protein structures, and the way to use that is to look into the InterPro database and look at the member protein signatures, and one or two of those do use protein structure in constructing their signatures, notably SFLD and CATH, the gene 3D. So if you want a signature which takes account of structure, you have to go and look in InterPro and see those.
So the question is, can we only run Unifier if we have Linux? So currently, Unifier is running on Linux, and it has also been well tested on Mac computers. So far, I'm not sure it can run on Windows, but if you would like to try it out in some Unix similar environment on Windows or even directly on Windows, we are happy to get your feedback about this, if you have succeeded and how you have succeeded. Then the next question is there a way to run automated interpro scan for multi-faster file locally and not using online interface? Yes, there is. So if you go to Interpro website, you will find the documentation how to install Interpro scan locally on your computer, and how you can then run Interpro scan on not only on one protein sequence, but a bigger set of proteins. Yeah, if you use the web interface for a bigger set of proteins, it may be a bit slow, but if you have 100, maybe 1,000 proteins to annotate, I would go for installing it locally. So you can download InterProScan libraries compiled, but InterProScan, I think it's a GitHub repository, is also freely available with the source code where you can compile it yourself and run it. So the next question is, when there are multiple rules that apply, can we determine which is the most specific rules? So the most specific rules. So I would say you can find the most specific rules by looking at the annotation that was triggered. So the more specific the annotation is, the more specific the case in the rule was. The next question is, does Unifier work with the residue level functional annotation? So yes, we have residues, so specific annotations which we call positional features, or I called in this webinar position-specific annotations. So like the two active site residues of the example case that I have shown, they have been predicted specifically for two certain positions in this protein. The next question is there a way to run a large FASTA file of non-annotated proteins and replace the header information in FASTA with Unifier export, such as protein name and gene symbol? So, basically we are providing a script where you put as input a FASTA file with one tax ID, and it will then replace this one with the full taxonomic lineage. This is what I have shown. But if you need if you need additional conditions, so you can add additional information like organism name to the FASTA header. So far there is no script included in the repository yet, but if you would like to have this, you could have a look at the scripts that are fetching the full lineage and extend it, that it also fetches something like organism name. So protein name and gene name are not used in conditions for Unifier. They are basically predicted information, and therefore you don't need to give this information in the FASTA header of the input file. So the next question is, Is UniFire specific to prokaryotic annotation? So it is, UniFire is not specific to any taxonomic group. So what the kind of proteins that UniFire can annotate depends on the kind of rules that you feed to UniFire. So in UniRule and SAS, we have rules across the whole taxonomic spectrum. So many rules are for prokaryotic annotations, but we also have rules for eukaryota or viruses and archaea.
Okay, I think we've reached the end of the questions there. So thank you very much for everyone getting involved. As mentioned at the beginning of the webinar, we will be making these slides and the recording of the webinar available, and which Herman mentioned will have his email address, and if you do have any further questions. A final slide from us. Please do go online and see what webinars we've got coming up. We will shortly be taking a summer break, but there's one more before we do, and then we'll be back in the autumn with a new series. Great, thank you, Herman, and thank you all for listening.
Thank you.